I always make many videos on the dissolution. Dissolution testing and dissolution testing topic is very big topic, and always we should try to learn regarding the dissolution. Dissolution is one of the most important, critical, and it is a performance test for the pharmaceutical formulations. I welcome all the viewers to this video on dissolution testing in pharmaceuticals, and this video will give you understanding regarding the principles, apparatus and regulatory aspects. This will be a very informative video regarding the basics. So dissolution, it is a laboratory test, you can say, or analytical test. It measures the release of the drug from the formulation. Dissolution is a laboratory test which measures how fast and how much of the drug is released from the formulation, it may be the tablet or capsule, into the dissolution medium. It simulates the process of drug release in the human body. That means it is an in vitro test which has direct relation to the in vivo drug release, and it helps to predict how the medicine will perform after administration. That means it is a in vitro test for the study of in vivo behavior of the formulation. Dissolution test is done for many of the formulations, whether they are tablet, capsules or oral suspensions, and sometimes these are also performed from solution formulations as well. Then the meaning of rate of release and extent of release is how fast and how much of the drug is getting released in how much time. Depending on to the formulation design like immediate release, delayed release, sustained release, modified release, the dissolution test is performed. Principle of dissolution. So in this test, the drug is released from the formulation or dosage form into a dissolution medium under controlled conditions such as temperature and rotation speed. The amount of the drug dissolved is measured at different time intervals to understand the rate and extent of the release. For example, you are working onto a dissolution test with immediate release tablet. You are putting the tablet in the dissolution medium. You are running the apparatus, it is paddle or basket, at certain RPM like 50 RPM for paddle and 100 RPM for basket, and you are testing the dissolution media for the drug release at different time intervals like 5 minutes, 10 minutes, 15 minutes, 20 minutes, 30 minutes and some other points like 45 minutes and 60 minutes. Now coming to the types of the dissolution test apparatus, mainly 4 apparatus are there used for the oral formulations like apparatus 1 basket, apparatus 2 pedal, apparatus 3 is reciprocating cylinder, apparatus 4 is flow through cell. These are used depending on to the formulation, like basket is used mainly for the capsules and floating tablets. Apparatus 2 is much more common than all the others, and it is used for the conventional tablets and capsules. Capsules, if used with paddle, the sinkers are used. Then short gelatin capsules are also tested with paddle. Apparatus 3 is suitable for delayed release formulations, non-disintegrating type of formulations. Apparatus 4, that is flow through cell, used for the powders and poorly soluble drugs. Now temperature for dissolution testing. So it is generally 37 degrees Celsius, tolerance of 0.5 degrees. The temperature represents the normal human body temperature to mimic the in vivo conditions or to simulate the body temperature. Understanding the Q value. So now the amount of the release is measured as Q value, denoted as Q value. The Q value represents the minimum percentage of drug that should be dissolved within a specified time. It is defined by the pharmacopoeia or monograph, the formulation development team, and approved by the regulatory authorities. For example, ATQ for immediate release type of formulations in 15 minutes or 30 minutes. Now, why 6 tablets are used for dissolution testing? Why 6 minimum? 6 is the minimum number of tablets. 6 tablets are used to check the uniformity of the batch according to USP. During stage 1 testing, each of the 6 units should not release less than the Q plus 5% of the drug release or drug dissolved. For example, if your Q point is 80 in 30 minutes, so all the tablets and average should be above 85 or equal to 85%. What is sink condition? So sink condition means that the volume of the dissolution medium is sufficient to dissolve the drug, and it is at least 3 times more than that what is required to dissolve the total amount of the drug in the dosage form. For example, the highest strength of the formulation is 100 mg. The dissolution medium should be such that it can dissolve at least 300 mg of the drug. This condition prevents saturation of the medium and ensures the continuous and complete drug release. That means it will reflect truly the formulation dissolution behavior. Degassing of the medium. So degassing is the process of removing the air from the dissolution medium, usually by applying the vacuum or heat. Then if air remains in the medium, bubbles can form and stick to the dosage form, leading to inaccurate or lower drug release results. That's why many of the formulations dissolution testing involves degassing. Coming to the calibration of the dissolution apparatus. So as like other instruments and apparatus, calibration is a important part for the dissolution test apparatus as well. Calibration ensures that the dissolution apparatus is working properly. It involves checking the accuracy of the rotation speed, verifying the temperature 37 degrees Celsius plus minus 0.5 degrees Celsius, and ensuring proper alignment of the paddles, basket, and using the USP prednisone tablets as a standard for calibration. I will make separate video on dissolution apparatus calibration and the performance verification. Method validation in dissolution. So as like analytical method validation, dissolution, it is a critical part of the method. Method validation for dissolution is required, and it is done as per the regulatory principles, ICH principles. Method validation confirms that the dissolution test gives the accurate, reliable, and reproducible results. And important parameters or method validation parameters for the dissolution test include specificity, linearity, accuracy, precision, robustness, and the filter compatibility. Filter compatibility is a very important part of the dissolution method development and validation. Regulatory guidelines related to dissolution are USP chapters are there, then pharmacopoeial informations are there. The USP, IP, EP refine the standards, methods, and their limits. International guidelines are there. ICH Q2 R1 provides the guidance for method validation. US FDA guidelines are there. ICH guidelines are there related to the bioequivalence, bioawareness, and stability studies. Dissolution testing is one of the most important and critical tests for the stability studies. Dissolution test is directly related to the safety and efficacy. That's why it becomes very important to understand regarding the dissolution. Now coming to the F2 similarity factor. So when you want to compare the dissolution of two different formulations, whether they are having the same manufacturer or different manufacturer, or whether these are compared between the reference formulation and the generic formulation or test formulation, that time F2 similarity factor is calculated to demonstrate whether the dissolution profiles of these two formulations are similar or not. The F2 or similarity factor is used to compare two dissolution profiles such as a test product and a reference product. If F2 value is between 50 to 100, the two profiles are considered similar. Now reporting the dissolution results. So complete dissolution report should mention the type of apparatus used, rotation speed, temperature, and medium composition. It should also include the percent of the drug released at each time point. Compare results with the specification and clearly state whether the product meets the requirements or not. System suitability test. So for dissolution testing, this SST is also performed. SST testing ensures that the entire system is functioning correctly before running the actual samples. This includes checking temperature, rotation speed, medium, and running calibration and running of the calibration tablets to confirm that results are within acceptable limits. Now coming to the example, like when one tablet fails, if one tablet fails to meet the criteria among the 6 tablets tested, additional testing is performed as per USP guidelines. So I have made some videos on stage 1, stage 2, tier 1, tier 2 resolution testing. I request you to go through those videos to understand the stages. For IR formulation, S1, S2, S3 stages are there. For MR formulations, L1, L2, L3 stages are there. In stage 2, 6 more tablets are tested, making the total of 12. The average of all 12 should be equal to or greater than the Q value, and no unit should be less than Q minus 15%. If still this stage 2 fails, then if the results still fail, go to stage 3, involve testing 12 additional tablets. Now you have 24 tablets, and then the overall average should be at least equal to Q, and no unit should be below Q minus 25%. Resolution testing plays a critical role, vital role in ensuring that the drug releases its active ingredient properly and consistently. It is a key indicator of product quality, bioavailability, and performance because it is an in vitro test to know the in vivo behavior. Bioequivalence bioavailability is directly linked to the dissolution. Following standard procedures and guidelines ensures reliable results and helps in obtaining the regulatory approval. So this is the information regarding dissolution. I think the video got quite lengthy, but it was necessary to include all the points. Thank you for watching the video, and I will request you to please do like, share and subscribe to this channel. Share these videos with your friends and colleagues. Thank you.